Okay. Thank you again for the invitation on this on the MVIF50. And I'm really happy to share our work on pollutant biodegradation in aquatic systems, specifically in rivers. So our group at Stockholm University, I'm a postdoc with multiple cell. We are really interested in the water cell purification of aquatic ecosystems. So this specific function helps with the basically cleaning of natural cleaning of aquatic systems. But there are many processes that are involved in this. And so we specifically focus on biodegradation, which are mostly microbially mediated. Specifically, how we measure this is by quantifying the ecosystem function using microbial transformation rates or biodegradation rates. And we are more interested not just on the persistent, but actually on the biodegraded pollutants, because at least from this, we could know if the ecosystem, the aquatic system we're assessing are actually naturally capable of self-purification. And to do that, we're actually using modified OECD 309 test, which is a standard test for measuring biodegradation rates in environmental samples. So to give you a very short background of how we do this quantification, basically we sample environmental sediments or water from aquatic systems and perform batch incubation assays. And with this, we spike it in the lab with a contaminant mixture. Specifically, we're using 129 organic micropollutants, which is represented by multiple use classes, for example, from pharmaceuticals, agrochemicals, cosmetics, and some food additives and industrial chemicals. And from this, using this framework, the group has already been able to present that biodegradation in aquatic systems, specifically in rivers, are seasonally variable. It is also spatially variable. The difficulty here is that because of this variability, it introduces major uncertainty in how we predict contaminant lifetimes, specifically in rivers. But this is expected because biodegradation, as I mentioned, is a microbially mediated function. So the variability reflects the fact that biodegradation is a biologically mediated process. So it is driven by microbial metabolic activity, and as we know, microbial communities are spatially and temporally variable. Also, community composition and interaction within species interaction can also influence the observed variability in biodegradation rates. But still, understanding biodegradation requires considering ecosystem-level microbial processes. To make this a bit more complicated, we also know that microbes are not just prokaryotes in the environment. They also exist in multi-trophic groups. So from bacteria, archaea, algae, fungi, and protists, and even metazoans. So this higher trophic level may influence degradation via predation, competition, and resource cycling. Still, we lack integrated studies that links multi-trophic interactions to biodegradation capacities. And so in this specific study, we aim to answer if microbial communities mediate the biodegradation of organic micropollutants in aquatic systems using our biodegradation rate framework, and specifically assess how multi-trophic microbial community shapes pollutant biodegradation potential in aquatic systems, specifically in rivers, and to quantify the extent to which this community structure mediates environmental effects on biodegradation profiles. So using previously collected sediment and water samples from Tian et al. That was published in ESNT, where she already have biodegradation data from 2 rivers in wastewater treated rivers in Sweden. We basically use those sediment samples and perform amplicon sequencing to profile prokaryotic and the eukaryotic communities from the sediment samples. And just to provide basic information on the biodegradation profile, we found that from the 129 compounds, 96 of the compounds had biodegradation rates, which means that those spike compounds in the environmental samples are actually degraded by the microorganisms in those samples. And they are spatially variable. So biodegradation profiles show clear spatial differentiation across river systems, which indicates that there is a structured variability in the potential of in the biodegradation potential of these test compounds. To link it with the microbial communities, we not just correlated the biodegradation profile with the microbial community, but actually perform mediation analysis to understand how these specific microbial communities mediate the influence of environmental effects on the biodegradation profiles. So exposures influence on the response, which is environmental factors on the biodegradation profile, and how the communities mediate this. And we found up to 60% of the total effect of prokaryotes on the influence of environment to the biodegradation profile of the two rivers, which is relatively high. We weren't expecting actually this much because in the natural environment, we know that there are other factors that complicates the biodegradation of specific pollutants in the system. And so seeing 60% of the influence of prokaryotes on environmental factors on the profile is actually quite high. Interestingly, we use functional annotations from the prokaryotic communities, and we found that all predicted functions and synobiotic, biodegradation, and metabolic functions have at least 30% of the influence. And this is sort of not too high since we're seeing 60% for the prokaryotes, but I should highlight here that these are annotated functions, so based on taxonomic profile. Still, prokaryotes are key drivers of pollutant biodegradation, and it captures a substantial portion of the environmental signal. Moreover, since we also profiled the eukaryotic compartment, we found significant indirect mediation by fungal, protistan, and even algal and metazoan communities. So fungi had strong indirect influence up to 56%. Specifically, this influences the composition, and some fungal species actually has enzymatic transformation processes. So they also have degradation capacities for certain micropollutants. So this is actually interesting, seeing 56% for the fungi compartment. On the other hand, protists specifically regulate bacterial communities through grazing, predation. So we found 53%, and this is probably because it's indirectly shaping the prokaryotic communities, so it has an influence on the biodegradation activity. And although not too high, we found 38 and 26% for metazoans and algae respectively. And this is most probably because these communities have an influence on the microbial structure via bioturbation and top-down control for metazoans. And for algae, they also contribute to primary production and organic matter supply, which has an indirect influence on the prokaryotic communities. And to highlight, we found that biodegradation is an ecosystem function shaped by spatial contexts and multi-trophic interactions, and that it is spatially heterogeneous. Contaminant transformation rates vary across river systems due to site-specific conditions, and that the rate constant we found are context-dependent. Single site measurements may not represent larger-scale environmental behavior, and specifically for the multi-trophic interactions, not just prokaryotes, but even multi-trophic groups regulate contaminant fate. For example, eukaryotic communities indirectly control biodegradation through food web dynamics, specifically by influencing the prokaryotic communities. And current risk assessment frameworks may be incomplete, so standard approaches often overlook community structure and trophic interactions. With that, I would like to acknowledge our group at ACESO, the Contaminant Chemistry Unit at Stockholm University, and our funding sources. And this study was recently published in the ISME Communication, so please do check if you wanted to know more. There are other analysis that I was not able to put in this 5-minute presentation, so please do check out this publication. Thank you so much.